reactions occurs, they're falling down, right? And there's a negative change in free energy, and the energy, this favorable energy, is being harvested effectively, and then those electrons are used to pump protons. And I'm going to show you how the proton pumping works. Not quite yet, but we're going to get there. Okay, but first, let's just look a little bit at how this actually works. So we have a combination of, this is a flavin mononucleotide here. These are iron-sulfur clusters that are in some of these enzymes here and here. We have cytochromes, and these cytochromes are molecules that bind very tightly to heme, and that's this structure that's shown over here. So here's a drawn-out picture of a heme molecule that has iron in the center. So this is, analogous. Hemes are used in hemoglobin to bind oxygen, but here in the electron transport chain, they're actually used in redox reactions to shuttle electrons. And these are called prosthetic groups. Remember, we talked about these in a previous lecture, but here you really see why we call them prosthetic groups. They're really intimately associated with these cytochromes. They're bound really tightly. In fact, we can't get them out, experimentally. If you try to extract heme from cytochrome, the only way to do it is to denature the cytochrome, right, and destroy it effectively to get the heme out. So they're not amino acids, right? It's a separate molecule, but it's considered to be, a prosthetic group because it's an essential part of the cytochrome. It doesn't come out unless we destroy the structure of the cytochrome. And so what happens here is that we have here's where we start to harvest, in a way, the energy that's stored up in these reduced forms of NADH and FADH2 in the form of these redox reactions where we can actually harvest some of the stored energy. So a molecule of NADH enters at the top of this electron transport chain, and it gets oxidized back to NAD+, and then this is a redox reaction that then triggers another redox reaction over here. And I want to point out something here, which is that this Q stands for coenzyme Q. It's also called ubiquinone. This is a molecule that's actually not embedded within the membrane. I shouldn't say that. It is, but it's not a protein molecule. So it's a smaller molecule that is more able to move around in the membrane. Right? And that's part of the way it works, is in sort of shuttling electrons between this big complex 1 and the adjacent complex 2. So that's a molecule that's moving around. It's not a protein, but it's really important. And so if you go to the store and you see, you're looking for, if you go to the section, vitamin section that says, are you tired all the time? Do you need more energy? One of the things they'll try to sell you is coenzyme Q, and that's actually what they're selling you, right? Is this ubiquinone that sits in the middle of this electron transport chain. All right, so electrons are transferred from NADH or FADH2 through this chain. They're passed through a number of these proteins called cytochromes that each have an iron atom embedded within this heme molecule, and eventually these electrons end up getting transferred to oxygen to produce water. And importantly, this chain is not generating any ATP itself, right? So back to this picture. So what's happening in this transport chain is that we're just basically harvesting electrons from these molecules in the process, but we're not generating any ATP directly. So you might be wondering how we're going to get ATP, and we're going to get there. But this is an essential part of this process of harvesting the energy that's been stored up in these molecules. Another important thing I want to point out here is that NADH enters this chain at the top, sort of in the highest energy state, right? FADH2 enters here at the stage of complex 2. So it's entering at a slightly lower free energy value. So there's not as much energy, effectively, that's stored up in FADH2 as we get from NADH. We'll see that in terms of ultimately the number of ATP molecules that are generated per molecule of FADH2 is lower than what we get from NADH. Okay, and we'll see that at the end. Okay, so we're not generating any ATP directly, and the function of this chain, again, is really to break the large free energy drop in going from molecules of food, effectively, molecules of glucose, to transport those electrons all the way to oxygen, breaking that up into smaller steps that release energy in manageable amounts that the cell can actually utilize. Okay, so it's a really important part of metabolism, is being able to do this. All right, so how do we get ATP? And this is where this process of chemiosmosis comes in. So this is really the energy coupling mechanism, right? And so what happens is that electron transfer in the electron transport chain causes proteins to not only to carry out these oxidation reactions of NADH and FADH2, but in the process, they also pump protons from the mitochondrial matrix, the interior of the mitochondrion, across that membrane into the intermembrane space in the mitochondrion. Okay, so it's very important. Here's where the whole directionality of these proteins and complexes come in, right? It's essential that they're oriented in the right way in that membrane so that they can pump protons in the correct direction. And so when they do that, then they set up a chemical and also an electrical gradient through the process of this kind of pumping. And then what happens next is that these protons can move back across the membrane, and they're now moving down both a concentration gradient and a voltage gradient, right? So that's a favorable process. And when they do that, they don't just do it willy-nilly or by random diffusion. They actually do it by passing through channels in this large complex called ATP synthase, which is a big, it's a big sort of machine that's sitting in the membrane. And these protons move across through a channel. And when they do that, this ATP synthase complex uses the favorable flow of protons down the chemical and voltage gradient. This is an exergonic reaction, right? It's a favorable negative free energy reaction that uses that process to drive phosphorylation of ATP. Okay, so we're phosphorylating ADP, adenosine diphosphate, we're adding an inorganic phosphate to regenerate molecules of ATP in that process. Okay, so this is a really beautiful example of chemiosmosis, the use of energy in a proton gradient in this case to drive cellular work. Good, so let's take a look at that. So this is a picture of the ATP synthase that's in your book, and it shows sort of a very cartoony kind of picture of this. So let's just get oriented. So here's the lipid bilayer, right? So this is that internal membrane in the mitochondrion. The matrix of the mitochondrion is over here, and then we have the intermembrane space on this side. So you can see how we have a really nice sort of directional process that's being set up by the way that the structure of this synthase is oriented in the membrane, and it's set up so that the part that's actually embedded within the lipid bilayer is literally a rotor. Okay, it's a rotor. It has each of these little blobs is actually a protein subunit, right? So these are each protein subunits that are sitting in this lipid bilayer, and there's literally a stator, and so these protons are able to enter a little channel, they enter sequential binding sites in these subunits of the rotor, and the rotor is actually spinning around, right? It's spinning around. These protons are entering. They spin this structure, and as they do that, they actually turn a shaft, this internal rod that is linked up to a series of catalytic subunits that are inside the mitochondrial matrix on this side of the membrane over here. And it's these catalytic subunits that are actually catalyzing the production of ATP from ADP and inorganic phosphate, right? And so these are the steps in this reaction. So these protons enter the channel in the stator. They bind to specific sites in the rotor, and they cause conformational changes in these protein subunits that actually act together to spin the rotor within the membrane, right? So this thing is spinning around, and each proton makes one complete turn before it enters the mitochondrial matrix through a separate channel in the stator. So it gets in there, it's like a merry-go-round. It gets in, it goes all the way around, and then it drops down through the rest of the stator, okay? And what happens when the rotor spins is that this causes this rod right here also to spin. And the rod extends into the stationary catalytic knot. So this part isn't spinning down here. This is actually stationary. But it's the turning of the rod that, again, through conformational changes, activates the catalytic centers in these catalytic subunits that then trigger them to generate ATP from ADP. Now, this is great to describe, but it's really fun to watch. So let's watch a little movie of this. Oh, let me like to play the sound here. So let's.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP.
Please turn down the lights.
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only pertonated subunits can then rotate into the membrane, away from the static channel assembly. Once the pertonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized.
This is a real structure right here, crystal structure.
Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
All right. So when ATP synthase really gets cranking, it can go at about 8,000 RPM. You can see that thing just, it's really cool. Okay. And by the way, I'll post that in the notes for today, so that if you guys want to go back and watch that movie or related movies, you can find it later. Yeah. Yes, so that's right. So because, again, it's a reversible reaction, right? And so like we've talked about with other enzyme catalyzed reactions, this is also an enzyme catalyzed reaction. So if we have a really high amount of the product, in this case ATP, it actually drives the reaction in the other direction. Okay, yeah. That doesn't usually happen, obviously, but it could happen. That wouldn't do any good, right? So that wouldn't help generate ATP molecules. We'd be burning them in that case. Okay, so basically, the energy that is stored in a proton gradient across the membrane is what couples the redox reactions of that electron transport chain to the process of ATP synthesis through that ATP synthase machine or motor. And we call the proton gradient, we can call it a proton motive force, right? A proton motive force that emphasizes its capacity to do work through this chemiosmotic gradient that's set up. So let's look a little bit, now the last few slides sort of show a little more schematically, kind of putting all this together, how we think, sort of imagine that this is actually working. So again, we have, here's that inner membrane, the mitochondrial matrix is in here. And here's intermembrane space. And here are these individual complexes of the electron transport chain, 1, 2, 3, and 4, that are involved in harvesting electrons from NADH molecules or FADH2 that enters here and transferring them through this whole series of reactions, ultimately to oxygen to make water. And so the gold arrows are basically just, showing the transport of electrons, which eventually pass off to oxygen at the end of the chain. And really importantly, what this cartoon is showing you is that for 3 of these 4 complexes, complex 1, 3, and 4, not complex 2, these are actually transporting protons to the intermembrane space. So these guys are the ones that are actually generating that proton gradient in the first place through the process of these redox reactions, right? So they're setting up the gradient that is then used over here by ATP synthase to drive the production of ATP in that very elegant little mechanism that we looked at with the rotor going around. Yeah. What's the purpose of making water? Well, it's just, oxygen is a really good electron acceptor, right? So it's just a way of having a really good electron acceptor at the end of the chain that accepts those electrons. Yeah. And So these brackets just show you that overall this process is referred to as oxidative phosphorylation, but it has these two components: the electron transport chain over here, where we're not actually directly making ATP during those redox reactions, and then chemiosmosis over here, which is where ATP is produced using the gradient that's set up during the electron transport chain reaction. So be sure when you study this that you kind of think about these different steps, where this is going on, and how we're actually getting from complex organic molecules like glucose all the way ultimately to ATP. It's clearly a series of indirect kinds of things that are going on, but that are highly coupled to each other to lead eventually to the production of ATP molecules. And so we can do a little accounting of ATP production during this process of cellular respiration. So basically, energy is flowing in the form of glucose. We're reducing molecules of NAD to NADH, or in one case FAD to FADH2, going through the electron transport chain, producing a proton motive force, and eventually generating molecules of ATP. Now, in the old days, I guess, or at least in the previous version of the textbook, the calculation was that about 40% of the energy in a glucose molecule is transferred to ATP. Now, your book now says, well, it's really more like 34%. Does anybody know how do we do that calculation? If you want us to do that calculation, we said, your book says, well, instead of 38 molecules of ATP being made in the process, maybe it's closer to 32 molecules of ATP. So how do we do that calculation? Yeah. Right. Exactly. So we can just combust glucose, oxidize it completely, right? And your book says, well, that if we do that, we get 686 kcals per mole, right, of glucose, right? So 686. And does anybody remember what was the free energy change during ATP hydrolysis, ADP, inorganic phosphate? 7.3, right? About 7.3 kcals per mole negative, right? So you can just do the math, right? So you can just calculate. So if we had 32 molecules of ATP being made times 7.3, we get, I did this earlier, 233. Right, 233.6, and we can just divide that by 686, and we get, closer to 34% efficiency or so. That's still pretty good, right? So a car engine, if you have an internal combustion engine, it's probably only about 25% efficient. So this is actually a pretty efficient process, it turns out. And then this is just showing you the overall cycle of everything that's going on. And in case anybody's wondering how do prokaryotes do it, they actually they don't have a mitochondrion, but they have two membranes, right? They have an inner membrane and an outer membrane. So they do the same thing through that inner membrane space between the two membranes that surround the cell. See you on Friday.